CRISPR, uses in medicine. Table of contents. First, we will go over what CRISPR is and hopefully gain an understanding of what CRISPR actually means and where it comes from. We will then go over the applications of CRISPR and how it relates to the treatment of disease. Next, we will talk about CRISPR in modern medicine. Then we will talk about the limitations of CRISPR and what can go wrong with this approach. After that, we will talk about the many ethical concerns that CRISPR brings. Finally, I will read a short concluding statement. I'm sure a lot of you have heard about CRISPR, particularly in regards to topics that were trending or recently trending, like its use in COVID-19 research. I know a lot of you also understand the premise of CRISPR, like how it is able to quote-unquote edit genes, but I'm willing to bet that there are at least a few new things that you can learn from this presentation that you didn't previously know about this topic, especially regarding CRISPR's use in medicine. Chapter 1. What is CRISPR? CRISPR stands for Cluster Regularly Interspaced Short Palindromic Repeats. It refers to the repetitive DNA sequences originally found in bacteria with spacer sequences in between these repeats. Scientists found that these DNA sequences were then transcribed in bacteria and turned into RNA. The RNA guides a protein that cuts viral DNA, or otherwise known as a nuclease, which provides protection against the virus. These nucleases are called Cas, which is short for CRISPR-associated. Knowing this, begin to think about how this may be applicable to genome editing. Chapter 2. Applications of CRISPR. In the year of 2012, researchers discovered that you could guide a Cas nuclease, the first and most common one being Cas9, to any sequence in the DNA and cut out a targeted part of the genome. Because of this, the implications of CRISPR are massive. This means that pretty much any DNA sequence could be edited, including pathological DNA sequences. Harmful variants of a gene can be physically cut by Cas9 and replaced with the healthy version of the gene. This includes anything from mutations to problems with gene expression. As of now, single genes are being targeted. However, it is possible in the future that we will be able to target multiple genes at a time. CRISPR has significantly reduced the time and expense of developing animal models and specific changes to the genome that researchers use to study disease. In this way, biomedical research has very much changed. Now, I'd like you to begin thinking about how this approach can be used in medicine. Chapter 3. CRISPR in medicine. CRISPR is currently being used in clinical trials for a number of different genetic diseases and their treatment. Some of the diseases that CRISPR is being used for include but are not limited to the following: inherited blood disorders like sickle cell and hemophilia, inherited eye disorders, muscular diseases like muscular dystrophy, genetic liver disease, congenital genetic lung disease like cystic fibrosis, genetic deafness, and neurological disorders like Huntington's disease, Alzheimer's, and Parkinson's disease. Additionally, on top of the aforementioned genetic diseases, CRISPR is also being used for the treatment of infectious diseases like viral infections, hepatitis, non-viral infections like fungal, bacterial, and parasitic infections, and even AIDS. Another use of CRISPR is in the treatment of lung and breast cancer, specifically in stopping the progression of the disease. Although CRISPR shows promise, there's still a very long road ahead of this scientific field, as almost all of these treatments are extremely experimental and will not see applications in human patients for a very long time. Think about what some possible limitations of CRISPR may be. Chapter 4. Limitations. There are many limitations of using CRISPR for genome editing, particularly those in humans. There are many off-target effects for one, which have been observed at an occurrence of about 50%. These are essentially unintended mutations or alterations to the genome that happen usually when Cas9 proteins bind to a sequence that closely resembles the target sequence. This can be dangerous for obvious reasons. If you accidentally edit a healthy part of the genome, you can possibly cause pathological effects. Another limitation of CRISPR-Cas9 is DNA damage toxicity. One other thing that can go wrong is DNA damage. A lot of the time, CRISPR-induced double-strand breaks can cause apoptosis, which is a huge safety concern for humans, particularly in cells known as HPSCs, or human pluripotent stem cells. Because of this, CRISPR edits are more likely to be successful in p53 suppressed cells, p53 being a tumor suppressor in humans, which means that there is a bias towards oncogenic cells in their survival. This can be dangerous for obvious reasons, because p53 suppression means cancer. Finally, the last concern I would like to talk about is immunotoxicity. This is a huge concern. It's pretty straightforward, but basically CRISPR may elicit an immune response against the Cas9 protein, which is potentially dangerous for humans being treated with CRISPR. Knowing what you know now about CRISPR and genome editing, what possible ethical concerns can you think of? Chapter 5: Ethical Considerations. There are many different ethical considerations when thinking about CRISPR, particularly with the future implications of CRISPR rather than its current day use. Many people criticize CRISPR because of its ethical concerns, specifically with gene editing. One of the most controversial parts of this emerging technology is the possibility of people using CRISPR in the germline cells, where the progeny of a person receiving CRISPR may be altered to favor a specific outcome. Not only is this criticized greatly due to the eugenic implications of this technology, where this technology could be used to get rid of disabled children like those with diseases that would be hard to care for before they are even born, but also for the possibility of CRISPR being commercialized for non-therapeutic gene editing to make designer babies like those with blue eyes and blonde hair. On top of that, we know from history that eugenics is not a morally sound argument to make due to a multitude of reasons. Another consideration is weighing the harm of possibly curing these previously incurable diseases with the risk of the previously talked about limitations and dangers of CRISPR. Is CRISPR worth the DNA damage, the off-target effects, or the immunotoxicity possibility? CRISPR could possibly cause harm to patients if the limitations of this technology are not properly addressed, which as of now researchers are in the process of doing this. However, it is still not perfect and CRISPR isn't 100% safe or without risk, at least in human patients, that is. Another problem with CRISPR is the possibility of this new technology remaining one that is, at least for now, only readily available to those who can afford it, which is not just or morally sound. This is a common problem in our current healthcare system, and until it is properly addressed by our government, this will remain an ethical concern. This next point is a little out there, but it still exists as a possibility and is a hot topic in regards to being something that people have discussed regarding CRISPR. It is the possibility that this technology could get into the wrong hands and be used as a means of bioterrorism. This technology is highly powerful when it is used correctly, but could be a great danger if somebody were to use it incorrectly. Thankfully, this is all theoretical, and nobody has used CRISPR to do such things. Finally, the last problem I will talk about with CRISPR involves particularly the editing of germline cells, and it essentially entails the fact that the embryo cannot properly consent to having their genome edited. Some argue that doing IVF or other natal treatments, particularly invasive ones, is also not respecting the informed consent of the fetus. This is also an issue as the therapy is particularly new and we do not know every risk involved with this technique. So it begs the question, can the mother and father even properly consent? However, the consent question isn't truly a consent question. It is a question of when the fetus becomes an individual, which is a slippery slope that I will not go into. However, this issue still remains one of the top ethical concerns when using genome editing technology. Chapter 6: Conclusion. The use of CRISPR in medicine is a nuanced subject that has many limitations and ethical concerns. Despite this, CRISPR-Cas9 as an upcoming technology in medicine is fairly promising and holds significant potential in revolutionizing healthcare as we know it. Here are my works cited, and that is all I have. Thank you for listening.